years, so maybe a little bit longer than that. My trajectory has been a little bit tortuous, but kind of came back full circle. So I'm originally from Mexico City, and I did my undergraduate studies at UC Santa Barbara in environmental science. At the end of my undergraduate studies, I actually worked for UC Davis as a field scientist. So it's really fun for me to be able to come all the way back to finally have a permanent position here at the end of an expedition that took me around the world. So after finishing my undergrad, I did graduate school. Well, I worked for UC Davis for almost a year, and I applied to grad school, and I did both my master's and my PhD at Cornell University in biological and environmental engineering. There I really began to work on subsurface systems, so specifically groundwater contamination. And when I finished, there was basically no job prospects in the US. So my advisor, who was Dutch, encouraged me to apply for postdoctoral positions overseas. So my partner and I ended up doing postdocs in Europe. So the first one that I did was in soil physics in the UK at the University of Aberdeen. And there it was a really nice way to move from biological and environmental engineering to really dealing with characteristics of soils that I was very unfamiliar with. Once that stint ended, I ended up joining the fluid mechanics group at ETH Zurich in Switzerland for a couple of more years for a secondary postdoc, and that equipped me with even more essential tools that I've now married into soil physics, fluid mechanics, and now going back to my roots of environmental engineering to sort of put together the research agenda that I'm leading here at UC Davis. So I've been here for 5 years. My group works very much in terms of fundamental science of how substances, fluids, and stuff that is in fluids move through porous media. We approach it from a very fundamental perspective. So even though I like to tell people in a general sense that we work on contaminant transport for groundwater systems, really at the core, we are fundamental scientists. And so it's been really nice to start the career here. Last year I got tenure, so it's been a really nice relief to move away from doing things that I know are almost guaranteed to work to actually starting to do more risky stuff. It's much more fun to be able to play around with things that are high risk and high reward and not worry about losing the job. So to me, it's been one of the biggest advantages of actually securing tenure at this point. And I've been very fortunate to come back to UC Davis. Cornell University is a land-grant university. It's the main one from New York State. And so my connection between agriculture was very, very strong. Here at UC Davis, this is a land-grant university of the state of California. And so it's been wonderful for me to be able to work with folks who are in the College of Agriculture. So I have a lot of colleagues in LAWR who I work with. And for sure, the USDA office here in town has been very much instrumental to getting a lot of what we do to have some pathway to apply to real problems, even though we are, as I said, we're fundamental scientists. But I guess maybe that covers sort of the extent of my history. I'm very happy to talk to folks about sort of what I had to do to get me to where I was going at every stage. It for sure has not been smooth at times or even clear that we were going to end up in academia. But yeah, things ended up working out pretty well, and I'm pretty happy here. I guess maybe a couple of things that I should mention is that I'm an NSF career awardee. I got the career in 2019, right before my son was born. And a year or so ago, I was given the Early Career Award from the American Geophysical Union, which is one of the biggest professional societies in our field. So that's been really nice to get recognition from peers about the stuff that we're doing. So I think with that said, maybe actually get started on the talk. So I can successfully share this. Now that we've moved to in-person instruction, we seem to have lost some of the skills on how to maneuver Zoom, but hopefully what you see is the actual cover of the talk. Marian? Okay, thanks. Great. So again, thank you so much for coming out to hear a little bit about what we do. So as Marian said, I'm going to be discussing some of the research that we worked on to try to untangle some of the nuances of how groundwater flows and how mass is transported within groundwater, but through structurally complex porous media. So before getting started on any science, I really want to make sure that I don't forget to acknowledge the wonderful group of people that I've been working with. So these are friends, colleagues, and mentors that I've sort of accumulated over the years, both in the US and in Europe, who've been just wonderful to work with. So I very much encourage anyone who's interested in diversifying their network to stick out claims in other places. So I have physicist Margot Dens, physicist Francisco Perez-Reche, my postdoctoral advisor at ETH, Marcus Bolzner, a very good friend of mine, Matthias Wellman at ETH, and another very good friend of mine who I barely overlapped with when I was in Switzerland, Joaquin Martinez Jimenez. They've been just fantastic folks to work with disciplines in geological engineering, physics, groundwater systems, fluid mechanics, soil physics. I've learned a lot from all of them. But of course, I need to give credit to the main students who carried out the bulk of the work of what I'm going to be presenting today. So I'd like to make note of Zoe Canavas, who's my PhD student here at UC Davis, Fabian Arns, who was a master's student with me at ETH that I tried to recruit to come here, but it ended up being a little bit difficult under the administration at the time. And Maxence Correll, who actually spent half of his PhD in Switzerland, the other half here with me in Davis when I initially started. So these guys have been tremendous about working hard and moving to totally different places to be able to work with me and achieve some great things. And of course, I would be remiss if I miss acknowledging the sources that are actually paying our bills. So the American Chemical Society, Petroleum Research Fund, the National Science Foundation, and the AXA Research Fund that funded a lot of the work that we did while I was still in Europe. Okay, so I guess a little bit of motivation of why we study flow and transport in porous media. As an environmental engineer, we are looking often at the main processes that are affecting the critical zone. And the critical zone, for those of you who are not familiar, is defined by the National Science Foundation as the entire layer on Earth that goes from the tops of the trees all the way to the bottom of groundwater systems. And within this layer, the biggest porous medium that we're concerned with is generally soils and rocks. And maybe this is a completely biased approach because I'm a hydrogeologist. But in this particular layer, the main interest is to understand how water is flowing. So namely, how fast and in what direction it's going, and how stuff in that groundwater is being transported. It doesn't necessarily have to move with the same speed and in the same direction as the water that's carrying it. So generally speaking, the stuff that we're referring to or that we're concerned about ends up being groundwater contaminants, and these pollutants arise as a variety of sometimes deliberate, sometimes accidental activities, but notably we can classify agricultural, industrial, and urban activities as big culprits for polluting groundwater and just water resources in general. As a society, often we try to agree on certain goals and try to manage as best as possible this natural resource, just proposing to, first of all, sustain use the natural system as a sustainable resource. So figuring out ways in which we can maintain not just the quality, but also the quantity of water available to the general public. We try to do what we can to prevent pollution. There's a lot of pollution already, but preventing any further contamination is a big one. And the final one is trying to do what we can to increase the success for remediation strategies. One big problem with groundwater contamination is that it takes a really long time to even figure out that you have a problem. And by the time that you do that, it's already been there so long that it's spread out very, very far in terms of space. And getting stuff out from underground is extremely difficult to do, if not impossible. And so we would like to make sure that whatever it is that we're doing is able to increase the success of remediation, which embarrassingly enough is very, very low at the moment. And our ability to essentially achieve these goals really hinges on two aspects that we don't entirely have a grasp on. So the first one is that we have a thorough understanding of the processes that are controlling how flow and transport phenomena behave. And the second one is that we have an ability or an availability of predictive models that allow us to delineate how quickly a pollutant is going to get to a certain location and how far its extent is. And so from a very classical perspective, so for instance, if you're taking my undergraduate course on groundwater systems design, you would learn that normal transport, so things that we would consider to behave according to the theory that we teach in classic approaches, would tell us that if you have, and what I'm showing here is an aerial view, that if you were to have a spill of some blue dye, let's call it, in this picture, right at the bottom of the page, that it would begin spreading in the direction of flow. So it would move in the main direction of flow, but it would start spreading both in the longitudinal as well as in the transverse direction. So it would spread sideways and also in the main direction of flow, to the point that it would acquire this oval shape. The color contours here are indicative of the concentration of that material, where the darkest blue is the most concentrated, and progressively it leads to fairly diluted concentrations at the very edge. This follows very closely the classical approach of how a material is moving according to concentration gradients. And it would give us the sense that we would have this oval shape contaminant that we need to deal with. Now, if we were interested, let's say your house were to be located exactly where the X is located, you might care to know how quickly that contamination has arrived in time at your drinking water well, and how quickly it's going to go back down because it's past your source where you're extracting water from. And that concentration signal, if we continue to assume that the system behaves in the way that we just painted it in a spatial manner, would also tell you that the concentration is rising in a very predictable way, and it would go back down in time fairly quickly, and with, at least in statistics, what we would consider to be a Gaussian distribution. So this is a nice, very mathematically simple way to define what happens in a porous medium, but it makes a big assumption that the complexity of the pore spaces through which this contaminant is moving are not distorting the shape of the plume. So this is what I'm going to be referring to as the structural homogeneity of the porous medium. But nevertheless, this is a classical view that is considered normal. Okay, normal mainly because it follows a Gaussian distribution in space and an almost Gaussian distribution in time. The reality is anything but this simple. So things that are much more realistic look a heck of a lot more like the picture that I'm showing here on the bottom, on the right-hand side, where again, I'm showing you an aerial view of a contaminant plume. This is a real one. In fact, it's reported by the National Research Council as one of the Superfund sites here located in California, where after 17 years, the contamination plume looks still very, very strong. What we've been doing in this particular site has been applying pump and treat remediation strategy, which is one of the most common ones to dispose, treat, or destroy contaminants that are already in the groundwater. But we can see that the contours are still fairly dark, and we have not reduced the footprint of that contamination. So I think you probably agree with me in classifying this as a very unsuccessful remediation strategy. But it allows us to learn a few things about what happens in reality. So during migration, as this particular material is moving forward in the main direction of flow, here it's also occurring upwards in the direction of the page. So as that material is moving, what we get is a very large concentration of that mass begins to break through in what are called preferential flow regions. So the main paths for flow. This gives us very quick transport of very concentrated material that is basically taking highways or shortcuts to get from point A to point B very, very quickly and with very little dilution. At the same time, what we see is that we end up moving some of that mass that was spilled underground into regions that have very, very low mobility in terms of how quickly that flow is moving or how quickly that water is moving. And that is illustrated here in the lighter contours. That material takes a long time to reach into these areas that are considered to be stagnant, but they get released back in time at very low but very significant concentrations. It creates this pervasiveness of contamination at particular at structurally complex sites that make it a very difficult way to a very difficult problem to treat, because these guys are moving back out or getting released into pathways that we can pump the water out and treat it ex situ at such a low rate that you, after 17 years of investing millions of dollars, you're still not even halfway done. But the combination of having preferential pathways and these flow stagnant regions trapping material and just releasing it back to main paths of flow very, very slowly, the combination of the two creates concentration signals that are anything but this very nice quick rise and quick fall in time that we saw earlier. In fact, you end up with a very skewed or a very crooked concentration signal that does not follow a Gaussian distribution anymore. And this really tends to be the norm. We've seen this manifest in a variety of settings, not limited to, let's say, experimental materials that we're constructing in the lab, so sand columns. We've seen this in field studies. We've seen that in natural pores that we pull out of the field and bring back into the lab to do some controlled testing. And we've even seen it in a very simple artificial media that is colonized by bacteria. So every single one of these illustrations, the important part is that you end up basically moving things very quickly. So the first time that you see a signal is earlier than what we would have predicted. And you continue to see a signal of that particular concentration for a very long time, where the x-axis in most of these plots is anyway a time variable. So you have this persistent release of some contaminant that you should be actually expecting in the system. For a lot of contaminants, getting the big chunk of it out is the most important bit. For many others, very, very low concentrations are so extremely toxic. And so figuring out at what point you will no longer have the problem of having water that is of too low of a quality makes a huge difference. And so being off by orders of magnitude in our predictions of when water is safe to drink again can be basically the difference between life and death of recommendations for environmental purposes. And so an important aspect that I want to stress is that the complexity of the pore spaces has essentially a domino effect in every single other process that we might care about that is happening underground. And so we start with a structurally complex medium. So for instance, here I'm giving you a 3D image of the pore space that is extracted from an stylitis limestone, the complexity of the medium cascades into having flow that is not uniformly moving through all the pore spaces. So this creates what is so-called flow channelization or preferential flow paths combined with stagnation regions, where some areas of the flow field or how quickly that water is moving are very, very fast and others are very, very slow. And so it's very difficult to classify this particular flow by a moving average when you have very large extremes on either end. But having this uneven distribution and complex patterns in the flow or how quickly that water is moving give us the so-called anomalous transport, where they were concerned with a concentration signal coming out. Well, because you have some preferential pathways, you'll probably end up having material coming out very quickly, earlier than you would expect. But you would also continue to see that concentration signal for much longer than you would expect if you didn't have this level of structural complexity. And finally, if the stuff that you're moving through that porous medium is reacting, then you might end up with reactions that are not uniformly distributed, but in fact are creating hotspots in space where most of the reaction is only occurring in certain locations of the medium. And that can be seen both as beneficial or detrimental depending on the process. But ultimately what we're interested in is understanding how the structural heterogeneity basically cascades into affecting the flow, the transport, and the way that things react. Okay, so hopefully at this point I've managed to convince you a bit that this anomalous transport is ubiquitous. We continue to see a lot of evidence coming out in the literature that this anomalous behavior is manifested basically at every scale that anybody has studied it. So whether your emphasis is in studying at the pore scale, at a core scale or a fracture scale, or even in the field, consistently we see these crooked signatures of concentration signals, both in time and in space. And so recognizing that, essentially thinking conceptually that the contaminants underground are moving as this very nice, very well-defined oval that is captured by volume averaging approaches is essentially one of the main culprits for us not being able to describe correctly what is happening in real systems. And so we really need better models that capture these features of early breakthrough and late tailing. And that's exactly what my team is trying to get at. So we are working on physically understanding the mechanisms behind processes that happen in porous media, where we're using physically based models that apply a probabilistic or a statistical approach to describe the behavior of what is so-called anomalous transport. We do this by putting together experimental, numerical, and theoretical tools to study both flow and transport phenomena in complex systems. And so I guess to be able to cover this in a fairly efficient manner, the idea behind everything that we do is to figure out how structure leads to flow, how flow leads to transport, and how transport leads to reaction hotspots. But we're working on it in pairs at a time. Hopefully by the time that I'm complete with my NSF career, we'll have a nice link that pulls all four dominoes together. And I'm going to be presenting the most recent ones that we've managed together. So for these types of structurally complex systems, there's kind of no easy way to be able to assess how quickly and how, I guess the flow distribution, the flow patterns that you would expect if you're moving, let's say, water through a structurally complex system, other than solving the flow with its governing equations in a numerical fashion. So for those of you who are familiar, we're talking about the Navier-Stokes equations, and solving this at the pore scale, so at the millimeter or even micron scale, is extremely computationally expensive. So So when my students Fabian and Zoe thought would be pretty logical would be to figure out, okay, we have a porous medium that resembles basically a network in 3D space. Is there any way whatsoever that we can relate the speed in which water is moving through individual channels to the geometry and the way that these channels are connected to one another? So what is called the topology in graph theory, so that we can figure out where preferential pathways are forming that are moving very, very fast, and where you might end up with stagnation zones of regions of the pore space that are moving very, very slow, and conceive of the problem as if it were resistance onto a graph. And so by approaching the problem this way, essentially what we wanted to do was to distinguish, okay, well, first of all, what do we consider a preferential path versus a stagnation zone when all you have is information about the local velocity of how the water is moving? And second of all, we wanted to understand where these zones of high and low flow resistivity form, by way of being able to connect that information to the underlying pore structure. The final question that we wanted to tackle is, well, why do complex patterns arise in the way that they do if all we have is information about the structure, which, by the way, is much less expensive to achieve than solving the flow numerically. And so the way that we ended up going about answering these three questions was to create a really rich data set of different granular material that we were able to extract with X-ray computed tomography. So for those of you not familiar with this particular approach, it's a glorified CAT scan, but for environmental samples or for samples in general. You take X-rays of your sample at 360 degrees, and a computer is able to put all those tomographs together or X-ray radiographs together to recreate a volume. And so the picture here on the left is a reconstructed volume of a granular material that we put together in a little cubic container. Tomography, by definition, gives you individual cross-sections of what, if you put it all together, renders a volume. And so we took each of those individual slices and used those as a unique sample. So we had thousands of these individual samples that we were able to use to understand not just how the flow is moving through them in this numerical fashion that I described that is computationally quite expensive, but also on the other hand, to extract from information about the structure, the pore network. And the idea is to sort of split the workflow into understanding how detailed flow behaves and how detailed structure is laid out, and figure out the link between the two. Because if we can skip the step where we're solving numerically for the very local velocities, it would save us a lot of time and would help make predictions a lot more efficient. And so with these thousands of data sets, the very first thing that we wanted to do was to establish a metric that allowed us to assess how channelized a flow is. Surprisingly, there is no well-established definition of where sort of a cutoff lies between what one would consider to be channelized flow versus not. And so we decided that maybe a binary approach is not the best way to go. So we came up with a metric that is on a sliding scale. And so it allows us to gauge quantitatively how much more preferential one sample is relative to the other. And this is because when you look at media that look very similar in terms of how big the grain sizes are and how big the pore space is, you could end up with flow that is very different between one sample and the other. And so our metric is based on percolation theory, and this helps us divide the flow or the pore spaces into areas that are very mobile, which are shown here both in pink and in white, versus the ones that are immobile or less than mobile, here shown in blue. Black on the right-hand side are the grains, white on the left-hand side are the grains, and the color scheme indicates the local velocity. So very briefly, this approach thresholds or divides the pore space into regions that are moving fast and those that are moving slow, based on how soon we were able to establish a connection that goes from the inlet boundary to the outlet boundary and establishes the very first connection. So by lowering the velocity in our system, we're able to figure out what is the slowest conductive velocity that is connecting our sample from inlet to outlet. And so this classification ends up being the very first step to at least be able to physically contrast and define where things are moving and where they are so-called not moving or fairly stagnant. And so one thing that we wanted to do is to be able to distinguish, okay, so how quickly are things moving when we're saying that they're moving versus when they are quote unquote stagnant. And so to be able to do that, we performed some statistics about the expected velocity in the white and the pink regions versus the blue regions in the back. And so that is what is illustrated in the graph at the bottom right, where we have the uniform sample where the flow is pretty much moving about the same in all pore spaces, here shown in blue. And then the preferential sample shown in red, where we have carved out a pretty clear preferential pathway that is going around the grains, like so. And so the hypothesis is that the difference in contrast between how fast things are moving when they're in preferential pathways versus in stagnation regions in the uniform sample is going to be very small. And we can see that in the plot below. So the blue graphs show us the probability density function that we're going to be moving at a given velocity if we're in the mobile region versus the immobile region, which are the dashed and the solid lines. I think respectively, it's very small, so I can probably read it on my screen. Versus the preferential sample, where if you're in the mobile region versus the stagnant region, the velocities that are being sampled are vastly different. And so this is not a surprise, but it's an explicit physical definition of what it is that you should be expecting if you're found in a mobile region versus an immobile one. So now that we've sort of gotten a sanity check of what we were doing, the next thing that we wanted to do is to begin extracting information about the structure, because we know that the pore space can be viewed as a pore network, which is what I'm illustrating here on the right-hand side. The white lines are the network that is created by the pore spaces in one of the samples that we worked with. And the idea was to be able to say, okay, so I have all of the points that are located on the far left are my inlets. All the points on the network on the far right are the outlets. Can we use approaches from graph theory to assess which is the path that is going to give us a match between structure and flow by just weighing a shortest path analysis to get us from inlet to outlet in the most efficient way, but sampling only pores of a specific geometry. And so Zoe thought that we might be able to do that if we were able to correctly ascertain the structural parameters that matter in terms of hydraulic resistance. And so by identifying the correct structural feature that gives you the answer that you already know is right, we're able to begin putting together not just the geometric features that matter, but the topological ones. So how these particular channels are connected to be able to say, okay, this is information about the structure that is equivalent to information about the flow. So she spent a good chunk of the pandemic in 2020 working on an evolutionary algorithm that tested everything that we knew about the pore structure to eventually be able to match not just what she figured out about the flow, but all the different candidate models that we had to be able to predict where that pathway was developed. And so this now allows us to just take structural information and say, well, based on 3 geometrical features and how the pore spaces are interconnected, we can tell you with very high certainty where your preferential pathways are going to be forming. And so that gives us now a better assessment of how transport is going to behave and where reactions are going to happen. So for this particular approach, we found that the most influential geometric features that are required to be able to predict this path of least resistance exactly where it forms is a ratio between individual channel lengths divided by the narrowest opening within them. And so we wanted, of course, playing devil's advocate, to assess how good this assessment was, not just for one sample, but we have these thousands of samples, all parameterized by the exact same model. But we wanted to have a control to compare how well we were doing. And so what we did was testing the accuracy of this overlap that I'm illustrating here between the pink region and this dotted yellow line, which is the model, relative to a control. The control that we ended up choosing was just the shortest path, counting the number of steps. I forget actually if this one is counting the number of steps or the actual length of the pathway. Whichever way this ended up being, I don't recall the details of currently. But what we see in the figure on the left-hand side is a complementary cumulative distribution function for the ability of our model, which is shown in yellow, to predict with high accuracy where these pathways are forming precisely relative to the control. And we see that in the way that one would read this graph, that for instance, let's say 90%, so this is in percentage of samples, at least 90% of the samples that we tested had an accuracy over 80%, or an accuracy of above 0.8 or 80%. So we're doing pretty darn well, particularly when you contrast it with our control, which, let's say this one will tell us that 90% of the samples are at least as accurate as 40%. So not doing so hot in being able to predict exactly where these preferential pathways are forming. And so we feel that this answers the where primary flow paths are forming or these preferential pathways, and what structural components are controlling where they actually form. And so this begins to shed a lot of light between the interactions that the fluid has as it's moving through these pore spaces and interacting with solids where there's an enormous amount of friction. The final question that we wanted to tackle was why? Why are these pathways forming at the locations where they are? Why is one porous medium channelized and another one is not? And so what we did was go ahead and basically break up the pore network into two subregions. So the first one ended up being the pink one, which is what we already defined as a pathway, the primary flow path. And we also took the blue one, which is what we're calling the stagnant region. And what we did, again, based on information or graph theory approaches, was to measure the level of order. So the so-called order and disorder is how convoluted that graph is. And namely, what we wanted to know is, okay, so we have this very strong preferential pathway. Is there some level of order that is found, structural order that is found in this pink guy equivalent, that is different from, let's say, the disorder that we're expecting in the blue graph? And the answer was pretty much yes. There's a very clear difference between the two. So what we find is that for graph theory approaches, a way to assess how ordered or disordered a graph is to measure its assortativity. So this R term is what we're calling assortativity. It's a preference that within a network, the edges or the branches that are making up the network, that they are attached to one another with some common feature. What we're arguing is the common feature is exactly that parameter that we use to define the shortest path. So this ratio between the length of the channel relative to its narrowest opening. And so what we ended up finding is summarized in the figure below. So if we look at one network with two subnetworks, we essentially find that the primary flow path has a very high level of order So this R coefficient is very large relative to its own sample's stagnant flow region. So every dot here is one of the samples. Each sample contains one piece of information for primary flow, one piece of information for the stagnant flow. And by and large, what we see is that the dots fall below the 1-to-1 line, indicating that there is a higher level of order for what is considered the pink regions, so the primary flow paths, relative to the stagnant ones. And so what we see here is that, okay, there is this structural order in the preferential pathways that is allowing flow to be channelized there with very little resistance. And the reason why flow is stagnant in the blue region is because you have a very fat channel connected to a very skinny one, connected to an intermediate one, connected to a fat one. And so the flow just can't handle this variability in resistance of moving fast and slow and fast and slow and fast and slow. It much prefers to just go consistently in pathways that are about the same size, not extremely wide, but consistently wide. And so this is a pretty exciting new way to look at how flow is being organized, and allows us to begin drawing some links between properties of the flow, which are very much dynamic, and relate them to static properties of the structure, which are, not moving at all, but are computationally much easier to obtain. And so the next, and I'll just talk about these two chunks of the research that we do. I want to talk about the last two domino effects. This is very, very recent work that we've been working on how this idea that you have anomalous transport is actually giving us a very non-uniform way of creating reactions. And so the specific reactions we're interested in are between a fluid and the rock. Specifically for this project, we're interested in dissolution. And so the governing question is, what are the controls for dissolution behavior in porous media, specifically with the outlook that this has applications for geologic sequestration of CO2 to combat climate change. And we wanted to come up with a model that is able to predict the reaction rates of dissolution in a system of arbitrary geometry or heterogeneity. So the classical view for understanding dissolution looks at the interplay between the rate at which things are being transported relative to the rate at which a reaction is taking place. And so dimensionless numbers are very common in engineering. So we often resort to this dimensionless number called Peclet that tells us whether we're flushing a system faster than we're allowing it to diffuse or dilute. Or perhaps we're interested in looking at it in dimensionless numbers for reaction, where either the Dampeler or the kinetic number essentially tell us that we're reacting faster than we're flushing things out. And so since we're looking at reactions between the fluid and the solid, it is important to sort of make sure that we are sticking to one of our two options for defining what the reactions timescales are. Just briefly, I'm going to be talking about the ratio between, are you allowing the reaction to happen quickly enough, or are you diluting it too much? And so here are examples of three dissolution behaviors that are commonly observed: so-called compact, uniform, and wormhole, where the porous medium is really the black stuff behind it. But if you're dissolving so well and barely flushing things out, you're essentially dissolving the rock entirely. So you have this compact kind of behavior. If you're balancing out how quickly the reaction is moving and how much you're flushing the reactant through the porous medium, you could end up with a uniform dissolution. So reaction is kind of happening everywhere throughout the system. And if you're moving things faster than they're able to react, you might carve out these so-called wormholes. And there are different reasons why you might prefer one strategy or the other. That is not necessarily of the highest interest to us. We just want to understand how the process works. And so there's very, very famous behavior diagrams that exist out there that try to relate these dimensionless numbers and the different types of dissolution behavior that exist, so that one can just very quickly read out a plot and say, okay, I am moving my fluid at this particular speed, and I know I'm reacting with some other reaction rate. I can back calculate my dimensionless numbers and at least get a back of the envelope idea as to whether I should expect uniform dissolution, wormhole dissolution, or compact dissolution. And these are really great, simple conceptual models to understand how dissolution works. The problem is that these kinds of dissolution behaviors are constructed from media that are very, very uniform in its structural features. And so most recently, when we've been trying to dump information into these diagrams of systems that are much more structurally complex than the individual gray samples from which the diagrams were made, we're finding that, okay, so the things that in the literature are reported as uniform fall in this upper quadrant, so that's really great. These are the blue circles. The triangles that are considered compact fall in the right quadrant, but we seem to be doing a really terrible job at classifying what is called a wormhole, or this channelized or hotspot reactions in the system. They seem to be given this apparent misclassification. And the question that immediately arose in our team was that, well, maybe there's other controls for dissolution that weren't missing from this particular diagram, because we're considering that things are moving at a particular rate and things are reacting at a particular rate. But we already know what having non-uniform flow does to the system. And so if we go back to basics and think about what is happening at the pore scale, so in between the individual pore spaces, we can begin to understand what it is that we might be missing from that diagram. And so I'm giving you a super, super high zoom in close up to a flow field that we've resolved. So these are, the black are individual grains, let's say of sand. The colors here indicate the speed at which the groundwater is moving in between them, and the white lines indicate the streamlines that material would move through if they were moving in these spaces. By way of dividing mobile versus immobile regions that I introduced before, we're able to say that, okay, of this colored map, we can say that the blue here is stagnant and the white spaces is where most of the material is going to be moving. Now, if you were concerned in, let's say, sequestering carbon dioxide, pressurizing it so it's in liquid form, and then injecting it underground for indeterminate storage, you might be interested in thinking about these individual molecules of dissolved carbon dioxide as these pink particles. Once you release them into the environment, they're going to be moving according to the streamlines that I just showed. Most of the material is going to move through the white part, and much fewer of those particles are going to enter the blue one. Right at the interface between what is considered mobile and immobile is we're going to end up accumulating these pink guys, and where we would expect the reaction to occur. And so if you pay attention to where the arrows are pointing, you can see through the animation that the presence of carbon dioxide, which creates an acid in the water, is ultimately what leads to the rock from dissolving. And this dissolution is not the same everywhere. It's going to be strongest where you're delivering most of that reactant. And so you begin carving out these so-called wormholes that we're not considering in the rendition that is classically studied. And so if we go back to our original apparent misclassification, essentially what we end up finding out is that we've been not misclassifying things, but looking at things from a two-dimensional perspective for a three-dimensional problem, where the third dimension that we weren't accounting for was this idea of flow arrangement, or how channelized that flow is. So this is what I'm calling the percolating threshold, or this PC term that we introduced in the previous project. Additionally, one of the things that we were able to ascertain is that the rate at which the rock is dissolving very much is in line or is related to the behavior that it follows. And so, for instance, rocks that dissolve by this idea of a wormhole behavior dissolve at a ratio that is much lower than those that are at uniform, and material that is dissolving as a compact is way off the charts. It dissolves at a rate that is much, much faster than either of the other two. And so because we have these correlations of now having an idea that the way that the flow is arranged matters, and that the way that the solution behaves is proportional, at least to a certain extent, or correlated to the reaction rate that we're expecting, we wanted to come up with a predictive model that allows us to tell what is the rate and what is the behavior that we should be expecting. So we chose a model that allows us to tease out of all the things that have been proposed in the literature, what are things that matter the most to be able to predict the reaction. So this is the expected value of the reaction rate that I'm calling RE over RB. The details of that are not important. So this tells us the rate at which we're going to dissolve that rock. We know that the transport rate, the reaction rates, this idea that we have flow heterogeneity or channelized flow, and a bunch of other things that have been proposed in the literature that we actually have information from in our samples could potentially matter, but we don't know exactly in what way and which ones matter the most. So from this vast data set that we put together, we're using statistical methods to tell us how these things are related, if they are nonlinear, and if there are interactions in between them, to be able to get at a correct parameterization of a model that tells us what the expected rate of reaction is. And so I'm showing here now the space map of our best chosen model. So we're able to tell according to how channelized the flow is and how quickly we're flushing the system out. We do a pretty decent job at predicting the ratio. So that is what is color coded here in between the blue and the red color map. And when we compare that against the samples that we've been training the model with, we see that we do a pretty decent job. So I'm giving here the same space. So the flow channelization is on the x-axis, and the rate at which we're flushing the system out is on the y. The circles are for each individual sample that is structurally complex that we're employing. And as the circles are small, that is proportional to the blue space here. As the circles get large, it indicates that the ratio is getting bigger, which is proportional to the red color here. And we do a pretty decent job between the two. So our model does a pretty nice job at predicting the rate, but nicely, we ended up finding out almost by chance that it also does a pretty decent job at predicting the behavior. So this is a qualitative assessment as to whether we're dissolving in the so-called uniform or so-called wormhole behavior. So we just relabel those circles, not just by the size, but by the type of dissolution that they behave in. We're able to pick up exactly what we're able to see in the model as well. So this is another pretty exciting view of using one simple statistical approach to get us two pieces of information that are in fact related to each other. Finally, we wanted to look back and say, okay, we have a system that is dissolving, and it appears as though if you start with fairly uniform flow originally, that you don't necessarily create these reaction hotspots. The more channelized we see that the system is, the more likely it is that we see wormholing. Is this related? And so we put that to the test. We have information about the progression of how the flow evolves as the system is undergoing dissolution. And so what I'm showing here is a plot of the initial channelization on the X against the final channelization of the flow on the Y. And so materials that are dissolving in the so-called uniform start very uniform and end very uniform. So this is nice. There are samples that start very channelized, so with very high PC numbers and end up pretty high. And that is the bulk of our samples. So most structures seem to start and end in what is called the same flow regime. So low PC initially, low PC at the end, high PC initially, or high PC at the end. which is what we deem to be reinforcing. So if you're not already redistributing things, then you're gonna, so however it is that you started is how you're gonna end. And that controls which behavior you're in. We do have some samples that seem to be doing the opposite. So they seem to be crossing over from being very channelized at the beginning and then somehow regulating themselves towards the end, or the other way around, which are here illustrated in pink. And we don't necessarily know why that is, but we're working on that at the moment. And so it's, again, bringing back information about how the structure controls the dynamics of the processes in porous media. And I think maybe with that, I will not take up additional time, but perhaps I just want to send you home with a few key take-home messages on processes that matter underground. The first one being that the structural complexity of the porous medium controls how the water is moving through it. So whether you're dealing with a system that is channelized or not, and this cascades into transports. So the delivery and movement of stuff in that water is very different than if it were uniform, and that catalyzes reactions in very specific hotspots within the system. And that is in fact the norm rather than the exception. The second point that I want to stress is that the topology of the flow, or how the flow is being organized within the pore structure, makes a huge difference in terms of how it's going to be arranged, but the order within the pores is what controls it. Finally, I want to emphasize that reaction hotspots are largely controlled not just by the reaction in the transport timescales, which is the classical view, but also how the flow arrangement is interplaying in delivering these reactants through either all of the pore spaces or just in a few of those. And so with that, I guess I would be happy to take any questions. I'd like to thank you for your attention and also invite you to either visit me here in the lab or consult on our website for the other exciting stuff that we work on, which is very much related to porous media, but various other questions that are pretty exciting and using cool, interesting tools to understand how little processes affect macroscopic behavior. But thanks.
Thank you so much for a fantastic talk. Are there any questions? Please unmute yourselves. And lots of clapping.
Thank you.
Any questions in the audience? And if not, I'll start. I do have a few questions. So as you know, my background is in mathematics. So I was very, and it's in what we used to label pure mathematics. I mean, I really don't like that label, but topology, geometry, graph theory, that's what I really enjoyed studying in college. So, and I still apply this type of models to DNA. So I was, of course, super interested in seeing and not surprised that the geometry and the topology are playing such a crucial role. Mm-hmm. My question, well, one of my questions is understanding the shape of the network seems to be very, very important. How much is known in general, depending on the material that you're studying, and how good are those models?
That is a great question. So there's, more network models are growing in popularity to understand flow and transport in the subsurface, but they are still very crude approximations of the real thing. I think the underlying question of how to create a random network that somehow resembles the real porous medium is very much still open. And so generally speaking, what I've seen our community do is, they'll measure the pore size distribution and ascribe that to the random graph that is being created. And maybe at best, incorporate the node degree or the local connectivity to the graph. And that's about it. And then we do simulations that have very little to do with real measurements in the real rock. And we're still unsure on what it is that's wrong with it. What we're suspecting from Zoe's PhD dissertation is that one big link that we're missing from that is this idea of assortativity. So the order that you get in a graph with the same size distribution and maybe even the same node degree distribution, but necessarily arranging them in such a way that even with the same global statistics, you have different pathways that are preferred or not by the flow.
Yeah, so you need to start thinking about the graph classifications themselves. Tell me again what the node degree represents.
So the node degree would measure at a very local level the number of connections that span out of a given node or a vertex. And they tell you intersections essentially within the graph. Are you connecting very locally between two channels that are crossed as an X, or do you have, let's say, a hub where many channels are connected by that particular node? So
it relates directly to the channels.
Yes.
I understand node degree from a graph theory perspective, but the edges are literally channels.
Mm-hmm. Yeah, in our case, they are.
Yeah, really interesting.
I'm gonna have to go, but I just wanted to mention that in the network they're modeled as channels, but the flow paths in the rocks are often surfaces with a width because they're the space between grains.
Mm-hmm.
And so I really appreciate also this network analysis for it. It seems like a really natural place, and I'm wondering, I don't know, I'm in earth and planetary sciences, so on the practical side, but I'm wondering if there's a way that to model the edges as being something more than a channel that might have I don't know, might give a little bit more realistic behavior or something of that sort.
Yeah, so for these models, they use, they don't solve the Navier-Stokes equation to resolve the flow field. They'll resort to Poisson flow in each of these tubes. So it's basically a bundle of capillary tubes. Mm-hmm. And so the equations for flow get simpler. You just need to make sure that they're balancing out and you have a divergent-free system still. The work that I've seen that adds a little bit more complexity to the individual channels is for fractured media, where the surface roughness of these particular openings makes a big difference in how things are transported, potentially mixed or retarded within individual chunks of the network. But that's as far as I've seen people incorporate additional information into these kinds of models. Mm-hmm. But it's not my forte because I prefer a different approach. We all fall into different classes if you're studying flow and transport, and pore networks are not my forte. I'm very interested in the link between structure and the dynamics of flow and transport, but I don't necessarily subscribe to the pore network approach. Okay. But thank you for your question, Don.
Thank you so much. I really learned a lot.
I'm glad to hear.
So you mentioned that you guys use an evolutionary algorithm to test what you know about pore structure. Can you say a little bit more about that?
Sure. I may need to call Zoe in since she's our expert on this. Let's see. So we're here. Essentially, what we are able to extract from these graphs is everything that we could think about measuring in terms of the geometry of the individual channels. So, the width, the length, how well they are connected. So let's say at this particular connection, you have 3 branches stemming from it. So we're throwing basically the entire pot of spaghetti at the wall and saying, this is everything we know about the system, but we don't know which ones matter to parameterize this shortest path analysis to tell us, okay, parsimoniously, these are the only two things that seem to make a difference so that you can still get agreement between the predicted shortest path based on resistivity and your known path based on the direct numerical simulations. And so that's what the evolutionary algorithm was trying to get at, is it shows a subset of the parameters that we think are important and started evolving them, combining them in different possible ways, in every plausible way, to see if we were getting closer or farther away from the prediction where this is the solution we know is right, but we don't know whether any of the models are getting better or worse if we tweak them a little bit. And so the evolutionary algorithm results I'm showing here on the left, I didn't talk about them at length, but essentially you take a first guess of what parameters matter, and you tweak them and you tweak them and you tweak them, and you compare against your known result. And if it's getting better, you keep those and you interchange them for other things until eventually the evolutions are stable and you're not tweaking them anymore. So that's how we were able to find out that this combination of length over pore throat characteristics of the geometrical features of a channel are the ones that really matter for the bulk of the samples that we tested.
And you also use that to, okay, so you use a genetic algorithm to predict the pathway and contrast it with the real pathway that you know
Right.
from solving the equations or from experimental observations, or both?
No, so this was numerical solutions to the flow.
Okay.
So we solved the Navier-Stokes
So
equation.
then you use a genetic algorithm to adjust parameters to see which ones give you the analytical solution, which
Right.
one matches the analytical solution. But that doesn't mean that if you did the experiment in one rock, it would be the yellow pathway, and in some other rock, it would be the blue pathway. I mean, even though like the optimal analytical solution doesn't mean that there aren't others that are close to optimal that by randomness would be explored in the field, right?
So what we're saying is that the most likely correct prediction just based on the structure would be based on the parameters dictated by the model here in yellow.
Mm-hmm.
And they would of course be a little bit different from sample to sample because they have different geometries. But provided that you know that one piece of information, you're able to extract the network of that pore space, you can do a pretty decent job at predicting where that pathway forms. And so that's why what I was trying to illustrate in this CCDF plot on the next slide is that, yes, we don't do a perfect job in every single sample. And mind you, these are thousands of samples that we tested all at once with that same parameter. Well, we can predict that pathway. So this is just one of thousands of samples. We can predict the pathway where you have the first breakthrough pretty darn accurately for the vast majority of systems. So we're going to be off occasionally, yes. But we do, it's a pretty darn good guess.
Yeah, no, that's amazing. That's amazing. Well, and my point here is that not that your model is off, but that nature is off, right? I mean, one thing is Navier-Stokes equations and the optimal solution, and another thing is what actually happens in the field.
Right.
So in that sense, of course you aim to match the analytical prediction, but it's good to sample statistically around the optimal solution, because when you go to the field, that might be the reality, that you're not getting the optimal solution, but you're getting something that approximates it.
Yeah, and there are certain structures that where the shortest paths and the second shortest path were very close together. And so let's say for the sample, this pathway and this one were almost the same resistance. It might be very difficult to tell experimentally if one of them was faster than the other when they're both breaking through at the same time.